Okay. Do you see the screen of a code?
Yes, we can.
Yes.
Okay, good. So we start. Okay, let's go. Okay, perfect score. So gram here, the gram is a gram staining gram positive, gram negative. It's a Dr. Graham that invented that technique. And Golgi is also a person's name. Sounds like Italian name. And Alzheimer is also other person's name. So that means if you write this word, you should start with a capital word, right? National name or the personal name. If it's a name, then you should start with the capitals. So if you write Golgi with a small g, that's a mistake. Okay, warming up is good. Second problem. Maybe the intention of the question was not very clear. Don't worry. I was asking about whether this is inside the body or outside the body. Okay, maybe the question was a little ambiguous. So blood or small intestine or stomach or surface of the skin, all those are outside of the body. Okay? Egg cell, egg is one cell. So inside the egg cell should be considered as inside of the body. Okay, this is asking about whether this is inside or outside. So this was a warming up. Similar question follows. Wow. Let's check one by one. Cytosol, is that inside or outside? Oh, you don't know what is a cytosol?
Inside.
Cytosol is inside or outside?
Inside.
That's inside of a plasma membrane, right? Inside of a cellular membrane. So cytosol is inside. Now, mitochondrial matrix, maybe you don't know what is a matrix. Mitochondria has a two membrane system, right? Double membrane. So outer membrane and inner membrane. Then inside the inner membrane is a mitochondrial matrix. The space in between we call intermembrane space. So then matrix should be inside. Intermembrane space can be considered as buffering space between cytosol and mitochondrial matrix. That should be considered as outside. Make sense? So then this is also inside. Lysosome. Lysosome is similar similar to endosome. When the endocytosis happens and lysosome vesicle is forming and then becoming more and more acidic, and this lysosome inside should be considered as outside, right? And how about the exosome? Exosome is the opposite of endosome. For forming exosome, what process do you need? Endocytosis or exocytosis?
Ectocytosis.
Not exocytosis. You need to have an ectocytosis. That's a different process I told you last time. Through ectocytosis, if this exosome is formed, then inside of this exosome is considered as inside of the cell or outside of the cell.
Inside.
That is inside, okay? That's why the other three, cytosol, mitochondrial matrix, exosomes are inside, and lysosome is outside. So this facing or topology is quite important to understand whether which side we are looking at, okay? One more problem similar to this. Okay, one person is not answering. Still, it's not clear. Now, cytosol is the only inside here, and vesicle is outside, and lysosome is one kind of a vesicle, endosome. And extended vesicles, you may consider Golgi or EL, those are kind of extended vesicle, okay? Then inside the Golgi, inside the EL, they are topologically, they should be considered to be outside, similar to lysosome, okay? So this other part is important for localization of the protein. That's why I tried to make some the underlining here. And later you will hear Sajin Sambe's talk considering this fact. Okay, not bad. What is the other name of rRNA?
Ribozyme.
Ribozyme is a little further extended name. Before you go to ribozyme, the direct name of rRNA is
Ribosome RNA.
ribosome, okay? Ribosome, zome means some kind of a small chunk, is a zome. Ribosome is the other name of rRNA. Ribozyme means RNA which has enzyme function. That's ribozyme, okay? So that's a little more abstract, bigger concept than ribosome. Ribosome is considered as one kind of a ribozyme. So is that clear?
Yes.
So usually we consider catalytic functions in our body. It should be done by proteins. Okay, those are we call enzymes. So protein, some of them is a structural protein which can build our body, but some other proteins which has a catalytic function, we call them enzyme. Later, we found that some RNAs, they have a catalytic function too. Then how should we consider those kind of new discovered materials? And the biggest one is ribosome, which is mainly RNA structure decorated with other protein. Ribosome is the center or machine of protein synthesis. And this is one kind of a catalytic RNA, and this is one kind of a ribozyme. Okay, there are many other RNAs which has catalytic function. Those are called as a ribozyme. Okay. Now, synthesis of rRNA is happening inside nucleus, and small area of the nucleus, so-called the nucleolus. In Korean, we call it in. That is the center of rRNA synthesis. Okay. How about the mRNA? Okay, not bad. mRNA is a messenger RNA. Maybe some of you may say, oh, we didn't learn it very well. Well, next week we are going to study about this material, but I assumed you have learned some basic study like this. Ribosome synthesis is done in nucleolus structure. mRNA is a copy of gene of the DNA, and the synthesis of mRNA is happening inside the nucleus. And then it is transported to outside. So the synthesis, the biological central dogma is information flow from DNA on genome, make copies of that information into mRNA, positive strand, and sense strand. So that's also we are going to cover next week. And then that information is translated into protein later. Okay. Protein synthesis is mainly done in cytosol. Cytosol is inside of the cell or outside of the cell?
Inside of the cell.
Inside.
That's inside of the cell. And usually this the protein synthesis machinery ribosome is attached to which organelle?
ER.
That's on ER. That's why if there are many ribosomes, ribosome is a huge the complex. That's why it can be looked as a small dots. So if there are many dots, we call them as a rough EL. That is the center of a protein synthesis. Now, depending on which direction they make this protein, okay, their final localization is decided. Okay, detailed story will follow later. Okay, so basically, the protein synthesis is happening inside of our body, in cytosol. Okay, inside of the cell. This level of question, I should expect 100% correct answer, just in case I checked. Gram staining is on cell wall, not or cell membrane, or Golgi body, or nucleus. So this part, we will hear Dongwoo's story later. Okay, I think this is the end of this piece. Okay, we move on. Okay, we just have done with this quiz, and there are several other things I wanted to cover. So this material was prepared by Sangwon today. Sangwon is not in good mood today, so I will use her material to present some part of her presentation. So nucleus is quite funny structure. Originally, it is not totally isolated structure. Okay, it is kind of extended structure from other membrane structure inside. So on a cell, we have plasma membrane, and then there's a small vesicles inside. Those small vesicles inside there, we should consider that as outside of the cell. Now, this extended membrane, it can be maybe kind of EL or Golgi. Then inside here, that should be considered as also outside. And those structures extended, and then they make this kind of a separate the structure, and they capture DNA inside here. Okay. Now, then there is some small holes here and there. This is the nuclear pore. And if you look at this structure, originally this was part of an inside, and just surrounded by extended membrane structure. That's why it has a double layer, one layer here and the other layer here. And this pore is kind of connected, continuously connected to cytosol. That's why inside the nucleus should be considered as inside of the cell. And a lot of DNA materials here, and also some small part of rRNA synthesis. Those are the ribosomal nucleolus structure. And then this nuclear pore is kind of a gate connecting this cytosol to the inside of the nucleus. Okay. Now this pore is a relatively big pore, and that's why small molecules can freely in and out. So that's why component in cytosol and inside the nucleus, they should be very similar to each other. That means we have high potassium here, then inside the nucleus we can expect there's a high potassium, not high sodium. Okay. So this pore is quite big, but it can filter out big molecules like macromolecules. They cannot go in and out freely. So those materials can be controlled by size or shape, but small molecules, they can go in and out freely. Okay, that's brief structure of the nucleus part. And this is the mitochondria part. So some of you may have the not clear the view of mitochondria. So outside here, this is a cytosol, right? And then this is one membrane. This is the outer membrane of the nucleus at the mitochondria. And then there's another membrane inside, bilayer the membrane. This is the inner membrane. So this is the inside. Originally, mitochondria we consider that's also came from separate bacteria. Maybe it has a one membrane structure like this. And they wanted to make interface between me and host cells. That's why they put maybe another membrane here. Then this space here, this space can be considered as this is outside of the cell. Okay, so this is a matrix. This is like original inner space of the original bacteria, and this is intermembrane space between two membranes. This is considered as outside. And in mitochondria, what is the major function of mitochondria?
ATP production.
ATP production is happening. ATP is energy material. That energy originally, where is the energy come from? What is the energy source? Is it carbohydrate? Carbohydrate was made by using which energy?
So
Light energy.
Yes, you should consider that as a light energy. Okay. Originally, photosynthesis is utilizing light energy plus very low level of the energetic material, CO2. CO2 provides carbon source, and utilizing light energy, they constructed bigger molecule and also the highly energetic molecules, so-called carbohydrate. So glucose is usually the first product, and then that glucose is stored energy. That stored energy is reconverted into energetic material, so-called ATP, utilizing a lot of these electron transport systems. So we didn't cover this metabolic process in details in this course for this year. We had other materials to cover, coronavirus and other things. We skipped that, but at least I wish you guys know that this inner membrane is active, ongoing energy generation, and also a lot of proton pumping processes ongoing. Okay, so proton pumping is happening here. They pump out the proton here. Then there's a lot of proton in between this space. Then you may consider this intermembrane space is acidic or basic.
Acidic.
High proton concentration, that means this is an acidic environment. Okay. Then how about the cytosolic space? Cytosol of our cell. What do you think? Should it be acidic or basic? Cytosol is inside of our cells, inside of the cell. Should it be strongly acidic, or strongly basic?
Should be neutral.
Yeah, I may prefer to be neutral. Okay, that is kind of too acidic, too basic. Both of them is extreme conditions. So cytosol is relatively neutral, slightly basic, 7.4 or something like that. So that's why most of those the cell cytosol, those are kind of near to neutral, slightly acidic or slightly basic, but it's not extremely acidic or basic. So then our cell is neutral. Then you may predict maybe this inside of a matrix. Matrix also, this was originally part of other bodies, bacteria inside, right? So that's why this is also kind of a neutral. Only this interspace, this is acidic. Similar things are happening in vesicles like a lysosome. Those lysosome is also quite acidic. So most of those outside space capturing inside our body, they are, you may think, they are kind of an acidic environment. Then you can, consider, or you can estimate inside of a Golgi or inside of ER, they can be potentially acidic if it is similar to a lysosome, right? So those are kind of outside environment. So that's another the aspect you can consider. Acidity and also maybe different ions. Outside here, a lot of potassium. Inside here, maybe higher sodium kind of things, okay? So this is how we can distinguish inside and outside of organisms in our life structure. Okay, one more thing that we are going to hear today. So I chose the two topics for you to present. So one of them is bacterial structure. Now bacteria is quite small, and one of these small rods, some bacteria is a globular shape that we call coccus. If this is a little long rod shape, then we call them the bacillus. And then the size of this bacteria is approximately 1 micrometer, right? And if the bacteria is a mixture of the different kind of bacteria, sometimes it's not easy to distinguish which one is which. So that's why about a century ago, there was one scientist named Graham, and he found the interesting staining material. And then he used some magic solution which stained different bacteria, and two kinds of staining. You can see very strong blue color and dark, the weak pinky color. So pinky color is kind of a counter staining. This is kind of a background staining, and this blue color is a real staining. So pinky color is, he called it, this is a negative, gram-negative bacteria. And strong color, this is a gram-positive bacteria. So this staining reagent, this is not antibody, this is chemicals. So he used some mixtures of colorful chemicals and then stained the bacteria and distinguished, at least made two classes, gram-positive bacteria and gram-negative bacteria. So very funny thing, the thing here is, it's not scientific classifications, just by adding some dyes, and if they are stained and gram-positive. If they are not stained, it's gram-negative. If Sejin found this kind of reagent, maybe this is SON positive and SON negative, maybe different kind of bacteria. Later, this stainings, the meaning, the structural meaning has been discovered. It has important the meaning and also insight of the structure. That part we are going to hear by the Dongwoo today. So first speaker for today is bacterial structure in terms of gram-positive and negative, and also the resistance to the antibiotic. Okay, that kind of a story we are going to hear from Dongwoo. And second one, I asked the Sejin Sanbe. This is a little more complicated the part. Whether this protein localization, how is it decided? Protein synthesis is happening in cytosol, but the final destination of each protein is quite different. They should go to the membrane and maybe inside the cytosol, or maybe delivered to different organelles like a mitochondria or lysosome, or some of them is used as a secretion, secreted to the protein. Then how the cells control that kind of localization of this protein, and you pay attention to each of them, whether this is inside or outside of those membrane structure. Okay, so those are two talks we are going to hear. So Dongwoo, you may start. Okay, Dongwoo, go ahead. You turn on your mic.
Okay, can you hear me?
Yep.
Oh, okay. Okay, so I'll start my presentation about bacterial structure today. And the four subtopics are like, the first is generally what a bacteria is, and the second is about the exact structure. And the third part is to classify the bacterials into gram-positive and negative as told by the professor. And the last is about health and disease, which means like the different kinds of diseases that bacteria can cause to us, and how we can resist to it, how the bacteria can be resistant against it. So that's the four topics we're going to talk about today. And the first to say bacteria.
Oh, you may make it to the slideshow.
Oh, okay.
Okay, good.
Bacteria is one of the two domain in prokaryotes, which is called as 원익생물 in Korean. And the other is archaea, which means 고세균. So bacteria is one of the two. And bacteria is very small, as we talked many times in previous lectures. And they're microscopic and single-celled organisms that thrive in very different kinds of environments. And they can live alongside with us inside our intestines or our guts and everywhere, or by lending helpers, or also to act as a pathogen which causes diseases like pneumonia. So these bacteria have different various types to classify, and the major four of them is each called as I can't exactly pronounce these four of them, but to do my best, it's Chylomyidias and spirochetes and cyanobacteria and gram-positive bacteria. So Chylomyidias is a parasitic bacteria that live with animal cells, and spirochetes or have helical structures, and they are called as heterotrophs, which means that they produce energy by eating and also using photosynthesis. And cyanobacteria, which are shown as green, are easily to assume that they make up their energy by photosynthesis. And the gram-positive bacteria is the main thing that we're going to talk about. So let's talk about it later. So the structure of bacteria is shown in above and has various structures. But the most important thing that we want to first notice is that all bacteria has a cell wall. And this cell wall has three important functions, which is to maintain the shape of the cell and to provide protection, whether it could be physical or chemical. It provides the protection of the bacteria. And it prevents the cell to burst or to get shrunken by hypotonic or very salty environments outside. So that's the major reason the cell wall exists. And this cell wall is made of cellulose or chitin, and so which has very structural. It has a very structural support. These cellulose and chitin has very strong structural supports. And bacterial cell walls contains peptidoglycan, which is a sugar polymer, which is cross-linked by polypeptides. So moving on, and the second most important features of bacterial structure is that they have no nucleus. So including us, other very much of eukaryotes have a cell nucleus inside, which is consistent of a double membrane. However, bacteria doesn't have it, and it consists of only one singular cellular chromosome. And this chromosome is not surrounded by a membrane, but is located at a nucleus region, which is like an ambiguous region that contains the chromosome like stuck inside of the cell. And these species of some species of bacteria also have smaller rings of DNA called plasmids. So this is very useful when we inject genes to bacteria to make insulin kind of stuff. So we might as well be focusing on that part too. And the other two features, it could be cool. These two cool features is that they have a flagellum and a pilus. These are both organic tails, I like to say, that maintains movement or to adhere, which means to connect to other bacteria to convey information or to other proteins which can make them evolve. So the reaction of the pilus is shown in here. So you can easily see that the pilus can connect to other recipients, and they can transfer information as well as other enzymes too. So so this is the features that we have talked about, and this next part is how to classify this bacteria. So this is a very famous experiment conducted by Graham, and I like to talk about how is the exact mechanism that divides this two bacteria into gram-positive and gram-negative. First, this gram staining is a staining procedure in bacteriology, which uses multiple staining techniques to divide bacteria into two categories, depending on their cell wall structures. Gram staining first starts with dyeing the bacteria in violet color or cross-crystal violet, or CV. Shortly after that, decolorizing with alcohol and acetone and counterstaining it with pink reveals its final color. If the color is pink, it's gram-negative, and purple when it is gram-positive. This color difference comes from the poor ability of gram-negative bacteria to retain its purple color. Purple stains only target the peptidoglycan layer, which is shown in here. However, the gram-negative bacteria only has a thin and unstable layer of peptidoglycan, whereas the gram-positive bacteria firmly holds the pink purple color because it has a thick peptidoglycan layer. Though the pink counterstaining in the end gives pink shades on both bacteria, the gram-positive bacteria tend to only show the purple color because it's just more attractive when the both are shown. The differences between these two gram-positive and gram-negative bacteria are very important when you're talking about antibacterial resistance. So to first know about what antibacterial resistance is, we have to first note that these bacteria can cause infections, numerous infections in our body. So I show an example on here, which shows that a strep Streptococcus and E. coli and various few other organisms can cause a flesh-eating disease, and it is very critical for our health to maintain and to keep away from these organisms. So I just brought this example as a kind of an example of an infection. And moving on, this antibiotic resistance, it happens when we want to kill the bacteria that cause infections, but when some bacteria evolve to have a rare featuristic to be resistant to the antibiotic. This means that antibiotics will not kill these resistant cells, and they can overtake the population to infect us in the second time gradually. So according to the CDC, which is the Center for Disease Control, almost 2 million people have been infected and 20,000 people have approximately died from the disease in 2013. However, this number has been astonishingly increased to over 40 or 50% in 2019. So that means that one infection occurs every 15 seconds, and one death caused by antibiotic resistance is happening every 15 minutes. So that means it's a very important disease, and something that we have to notice on, and we have to continuously focus our researchers on. So gram-negative bacteria, which is the main thing that we're going to talk about antibiotic resistance, is that they have less peptidoglycan and therefore an outer membrane that can be toxic. So this means that most of all antibiotics targets the peptidoglycan layer, whether it could block the synthesis of the peptidoglycan layer, which we have talked about in various weeks ago. However, gram-negative bacteria doesn't have much of this peptidoglycan layer, which means that they could be easily likely to be antibiotic resistant by all these penicillin and ampicillin and all these kinds of stuff. So I gave an example of this to American institutes such as the Congress or the CDC, that they're funding millions and millions of dollars over the past years that they're trying to overcome this disease, but it is actually a continuing disease that we have to always focus on. So this is the end of my presentation, and thank you for listening to my lecture. Can you hear me?
Sorry, I didn't turn on my mic. Sorry.
Oh, yeah.
Okay, thank you, Dr. Dongwoo. So could I invite questions from the class?
May I have a question?
Sure.
Yes.
During the exchange of genes in plasmid, what happens to flagella? Was it correct? Or when I saw the picture, it seems like flagella disappeared and reappeared again. So is there any mechanism for controlling about them? Please tell me.
Wait,
1 second. I'm moving there. This page, right?
Yes, it is.
Okay. Could you
I
explain?
didn't quite understand the question, so could you please repeat once more?
So during the exchange of gene in plasmid, the picture in number 3 shows like flagella disappeared.
Okay.
So does it have some mechanism that controlling the length of flagella, or just not seen in the picture?
Yeah, the picture looks like there's some flagella is involved in this exchange. Is that the case, or the picture is misleading us?
Well, I haven't exactly researched about that, but I think that the Pilus is kind of degrading and like generating again if I see this picture 3 and 4, but I don't know the exact mechanism. So
maybe the question, we can simplify the pilus and the flagella is the same structure or different structure?
Oh yeah, the flagella and Pilus is actually a different structure. So flagellum, which is shown in the left, is actually a tail of a bacteria which makes it move around. However, this pilus is just only a small substance of proteins that can be connected into bacteria numerously inside the outer membrane. So it's kind of different. The flagella and the pilus is different.
Yeah, two different structure, but with the simplified pictures, it just confused us, I think. Okay, other question? Okay, if not, let me point out here. Now you see, this is a peptidoglycan layer. This is gram-positive staining, gram-positive bacteria. And then inside here, this is a plasma membrane. Now, gram-negative bacteria, they have one membrane inside, another membrane outside, and then in between there's a thinner peptidoglycan layer. Okay, so by structure, you may consider which one is more similar to mitochondria. Gram-positive or gram-negative bacteria? Which is more similar to mitochondria?
Negative.
It should be gram-negative, right? Now that means when you say membrane, okay, this membrane is lipid bilayer structure. Okay, in virus we call it a little different name as an envelope, but that's also with membrane and then with some protein embedded inside. Those were considered as an envelope, a little different name we called. But here when you say membrane, these are lipid bilayers. So gram staining process was you add this violet dye first, so-called crystal violet, CV. And then in the beginning, maybe seems like both of those bacteria seems like a stained. It's kind of just attached. And then it's washed out with alcohol. Then when the dyes CV is binding to this peptidoglycan structure, it's strongly bound and it's resist to be washed out. But if it's attached to this membrane structure, then it can be easily washed out by the ethanol treatment. So then we can say that now we know that crystal violet binding site is this peptidoglycan layer, not the membrane layer. And the reason why the gram-positive bacteria is strongly stained is the dye directly binds to this and difficult to be washed out. And second of all, in this case, the dye cannot penetrate efficiently of this outer membrane. That's why it cannot reach this peptidoglycan layer. That's another reason. There are two different layers of membrane. And last time we talked about drug absorption. And there was the admit we talked about. Drug absorption is depending on the hydrophobicity or lipophilicity of the material. That's why there are two layers here, one layer and then two layers. If gram-negative bacteria is resistant to the staining material, same way it is resistant to antibiotic molecules, the drug molecule. Drug molecule should pass through one layer, second layer. That's why it is more difficult to go inside to the bacteria. Okay, that means gram-negative bacteria is usually more resistant to the antibiotic and do not allow the antibiotic the access to the inside material of the bacteria. Okay, so that's why some drugs, let's say we called penicillin and its related beta-lactam compounds. The target is here, peptidoglycan layer. So that's why it's a kind of expose the target. But if penicillin reaches here, if it cannot penetrate the first membrane structure, it cannot attack these peptidoglycan layers. That's why the penetration pharmacokinetic parameter is important, which has full access to this gram-positive bacteria, but they have difficulties to reach this gram-negative bacteria. Okay, so that makes a big difference of a choice of which antibiotic we should use. Another target Another the important class of molecule we discussed was the vancomycin. Do you remember what was the target of vancomycin?
Peptide.
This is-
peptide.
This is also the peptidoglycans, the peptide part, okay, part of this peptidoglycan layer, okay. So if you think about the cell wall structure, cell wall versus cell membrane, which do you think stronger structure, cell membrane or cell wall?
Cell membrane.
So
Now we have two different opinion, whether cell membrane or cell wall. I will give you a chance to argue about it. Is it cell wall or cell membrane? Which is the stronger structure? Let's think about the material. Cell membrane is composed of lipid bilayer, right? And the thickness is about we talked about this several times. Say again?
10 nanometers.
It's much less than 10 nanometer, about the 4 to 5 nanometer range. It's quite thin at the layer comparing to the big the cell size. Even the bacteria the size. Let's say bacteria size is 1 micrometer, right? If you make it million times bigger, 1 micrometer becomes 1 meter, right? Then 4 nanometer is, how thick is it?
4
millimeter. Then you can think 1 meter size of bacteria, and those cell membrane is only 4 millimeter. It's very thin, isn't it? If you capture those big ball, 1 meter size of a big ball with a so thin layer, that's quite thin. The membrane layers. The membrane is quite thin. Cell wall can be quite thicker and stronger, but you can consider cell wall is something like this. Do you see the picture? Kind of this kind of the cross-linked the armor. And if you look at those structure, okay, there is, let's say one line is the peptide, and one line is a carbohydrate polymer, and carbohydrate polymer is this x-axis, and then maybe y-axis is kind of a peptide, the linkage. They make physically very strong structure, physically, but there are a lot of holes there. Okay, that means this provides physical strength to the cell membrane, covering those cell membrane from outside. And this cell wall make that surface is more rigid, but the penetration of a small molecules through this hole is freely accessible. Small molecules can easily access through this big space. Okay, so this is similar to, let's say, nucleus pore, and any materials, small molecule, they can go in and out here. But the membrane is thinner, but it is tightly closed. It's not open structure. Even water cannot freely penetrate the cell membrane. Okay, then you can think gram-positive bacteria, they have this cell wall outside, just one layer, but thick one. And the inside there is coated membrane structure. Gram-negative bacteria, they have one lipid layer outside, and then this kind of the cell wall, even though that's thinner, but there's some peptidoglycan structure inside, and then another layer of the cell membrane inside. Okay, that's why gram-negative bacteria is more resistant to many antibiotics. Okay. Okay. Any other question for Dong's presentation? Okay, let's say it's clear for now. Then let's move on to the second talk. It's Sejin's protein localizations. Sejin? Okay, we can see the screen.
All right. Well, let's talk about protein targeting, and sorry for the noisy because I'm in a snack bar. So when it comes to protein targeting, these three are main questions you need to know, I think. So what is protein targeting, the definition, and where is the designated places for proteins, and how does it work? How is it determined? So actually the protein targeting, or we call it protein sorting, is a biological mechanism by which protein are transported to their appropriate destinations in the cell or even outside it. To determine the destiny of protein, signal peptide should control their location. Signal peptide is the piece of information that enable the cellular transport machinery to correctly position a protein inside or outside the cell. This information is contained in the polypeptide chain itself or in the folded protein form. Generally, signal peptide is found at N-terminal extensions. Some signal peptides can be at the C-terminal. Anyway, these signal peptides are recognized by specific receptors that manage delivery to the appropriate organelle. So here is overview of protein targeting. As you can see here, the translation of most proteins, even in a eukaryotic cell, begins in the cytosol. As a protein is made, it is sorted through largely two pathways. Among them, today we will focus on four possible destinies. One is following these passages. One is secretion out of cell. Other is embedded in membrane. These are categorized as a secretory pathway, or we call it co-translation or translocation, because translation and translocation occur at the same time. The other two is cytosol and mitochondria. These are categorized as a non-secretory pathway or post-translational translocation, because translocation happens after translation is done. So here is scheme. The protein is made in the cytosol first, and follow this blue arrow, it can be translocated to mitochondria or plasmid membrane or ER. After that, after some modification, the proteins are gonna move out of the cell following this vascular transport we will talk about now. So if proteins have a signal peptide on its own in the N-terminal, they are fed into the ER during translation in case of secretion protein. As a ribosome initiates translation, the signal recognition particle called SRP bind to the signal sequence on the N-terminus of the growing polypeptide. Once SRP bind to the signal, the ribosome halts the elongation of polypeptide, and then the ribosome SRP complex is then targeted to the ER membrane by interaction of the SRP and the SRP receptor. Next, the SRP dissociate the ribosome and signal sequence are transferred to the channel-like protein called translocation. Elongation of polypeptide chain by the ribosome forces the growing polypeptide through this channel. After the polypeptide is translocated through the translocon, the signal sequence is cleaved by signal peptidase, and the nascent polypeptide is released into the lumen of the ER. Once it is modified in the ER, I told you, it will be sent to Golgi apparatus by symbiotic and then secreted out of cell. I don't handle the detail of this vascular transport in this presentation because I contain too many content today. So I want to focus on this transmembrane transport, how it is recognized and how it is translocated from cytosol to specific membrane. And in case of membrane protein, a membrane protein's complete translocation by a slightly different mechanism from the secretion protein. The growing polypeptide is translocated through the channel until the stop transfer sequence is encountered. The stop transfer sequence then initiates release of the transmembrane polypeptide into the ER membrane, like this, through a lateral gate in the translocon after the signal sequence is cleaved by signal peptidase. This membrane protein and secretion protein are representative case of co-translational translocation. On the contrary, proteins that do not have a signal peptide, like this binding to SRP, stay in the cytosol following this pathway for the rest of translation. If they lack other specific address labels, they will stay in the cytosol permanently. However, if they have the right labels, they can be sent to the mitochondria, chloroplast, or nucleus after translation is completed. So this is just cytosol protein pathway is simple. This is kind of complex. If the protein has a mitochondrial targeting signal, chaperone called HSP70 bind to polypeptide, keeping protein unfolded after synthesis in the cytosol. All right, I'm a little bit confused. Right, then the chaperones deliver proteins to mitochondrial channel named as TOM first. It is an acronym of translocon outer membrane. It is translocon intermembrane. So the polypeptide encountered, this targeting signal encountered the TOM. The HSP70 molecules detach as polypeptide passes through membranes by using the ATP energy. And then the transit sequence cleave is similar mechanism. After that, mitochondrial HSP70 molecules rebind and release the polypeptide as it enters matrix inside the mitochondrial matrix. And then using this, the chaperone polypeptide fold by using ATP again. This is another picture to describe the same mechanism. So in summary, in the cytosol, the nascent protein is translated, and then if they have the signal of peptide binding to the SRP, they're going to follow the secretory pathway. It can be secreted out of cell, or it can be embedded in the plasma membrane following this biological transport. Or if it doesn't have the signal peptide, it can stay the cytosol permanently, or it can get the specific targeting signals. It can be transported into the mitochondria or nucleus or a chloroplast. We don't handle the passage of this nucleus chloroplast. It is quite complex. Yeah, anyway, this is the end of my presentation. Thank you.
Okay, there are a lot of information here. Maybe some part of it may not be very clear for you. May I invite your question?
Can I ask a question?
Sure.
Yes.
Okay. I have two questions, and the first is that on the chaperone part of HSP70. So
HS70 part, 1 second. Let me do it. Sejin, 1
Okay.
second.
Oh, yeah, okay.
This page?
yes.
Okay.
So what I'm curious about is if what I've understood is correct. It seems that HSP70 attaches to the protein 2 times before it enters and after it enters. So I can assume why it attaches after it goes inside the membrane, because it needs to be folded, but I can't exactly know the reason why it has to be attached before it goes inside. So I want to hear an answer about that.
This is my
Sejin,
question.
can you handle?
I need your assistance.
Okay, what is your TOM? What is your TIM?
The translocation of outer membrane and translocation
Okay.
of inner membrane.
That's outer membrane and inner membrane. Okay, let's handle the easy other one first. So let's think. Now here is the first the general case the first. This is ribosome. Everybody see my arrow, right?
Yeah.
Yes.
Okay, this is ribosome. So small unit and big unit, two ribosome is connected together, and this is mRNA. This is reading the information. So mRNA, there's a direction from 5' to 3' direction. It is reading that information this way. And then this is a synthesized peptide. By reading this information, peptides start to be synthesized. Protein is synthesized. And if this happens in cytosol, maybe this ribosome is attached to the surface of ER, or maybe just a free ER, the free the ribosome, floating in cytosol. Then synthesized protein will reside in the cytosol, right? So then that's maybe the simplest version of protein synthesis. Protein is just synthesized and secreted. This is synthesized and secreted. That's it. Then it can be just a cytosol. Now, if the protein has some information, in this case, Sejin said something about, so this is protein. Direction of the information of the mRNA starting from the 5' to 3' direction. Synthesis direction, it starts from N-terminal, amino acids N-terminal, amino acid is an amino acid, right? And it's connected to 1 by 1 here. So one side is amino group, the other side is the carboxylate group. So this side we call N-terminal, this side we call C-terminal. Starting of protein synthesis happens here first, and then it make long chain of peptide or the protein. The details of this protein synthesis and also RNA synthesis will be covered next week at the material. Translation and transcription you will study. Anyway, today we just simply say there is a direction from 5' to 3', N-terminal to C-terminal is synthesized. If this ribosome is synthesizing this peptide, then without any other functions, maybe this protein is apt synthesized. It is folding into active structure. During this folding, we need some kind of a helper protein, so-called a chaperone. Chaperone means it's a kind of a carrying helper who introduce new girl to the dancing club, kind of an old woman who carries the new girl to the dancing group and introduce to everybody. That is the original meaning of a chaperone. So this chaperone, originally, this peptide is just synthesized, not properly folded. Chaperone helps those folding. That is a chaperone protein. Okay. Now, anyway, if this protein is just synthesized and nothing much to do, then they will reside in cytosol. That's it. Let's make it simple. Now, if this protein needed to go to the mitochondria or needed to go to the lysosome, some specific organelle, then they carry this kind of specific signal, so-called the signaling peptide. And this signal is recognized by other shuttle protein. This signal does not need to be long. It can be 2 peptides, 3 amino acids, 4 amino acids, kind of a short peptide, which can be recognized by shuttle protein. They bind to this, and this shuttle protein carries this protein to the target. Then it can carry the target to, let's just say, mitochondria. This is sequence of a signaling sequence. Then this carrying molecule bind here, it carries into the surface of mitochondria. That's how those destination is decided by this signaling molecule and helper by shuttle the protein. Other protein carries this to this structure. Okay. Now here, this receptor can be maybe that kind of a shuttle, and then bind it here and then carry that onto the surface of mitochondria. Before we come back here, then the protein can be delivered to the final target of mitochondria, lysosome, or nucleus, or different structures. Now, another one for secretion. If this ribosome is attached on the surface of this ER, and there is another which helps those connection of this material into inside of this lumen of ER, then synthesized protein is secreted not to the cytosol, inside of this lumen. Then the final product is residing inside ER. Okay. In the case, now this side we call cytosol is inside of a cell. Inside of ER is inside or outside?
Outside.
That's outside, right? That's why this is important. That's why I tried to repeat those questions whether this is inside or outside. Right now, this ER looks like a little long membrane structure, but if this is forming like ectocytosis, it is forming this kind of vesicle, capturing this one, and if you disconnect here, then this whole ball vesicle can move too slowly to plasma membrane and fuse to the plasma membrane. That is what we call exocytosis. If exocytosis happens, this membrane structure fused to the plasma membrane, inside the material is secreted out to the outside. Do you follow me? This is how those proteins can be secreted. It's already it's moved to outside the space, and then this whole space is moved to the plasma membrane and fused, and then just pour out to the outside. Okay. Now membrane structure, maybe similar things is happening. The product is moving to the outside of the space. Whatever happening in between, some part of it, does not move fully inside, and some part of it is hanged on the membrane. Okay, this is kind of a half and half, let's say, C terminal here and N terminal here. If this whole structure, now this ER structure is moved to the plasma membrane and then fused onto plasma membrane. Because of this binding on the membrane, they are not secreting, but they are reside on the plasma membrane. Now the key question is, N terminal versus C terminal, which is exposed to the outside?
Intern.
Is that N terminal?
Both.
Both? Hey, here this is membrane structure. This whole structure is moving to plasma membrane. Then it is also should decide one side should be inside the plasma membrane, the other side should be outside the plasma membrane, right? And if you compare this with the previous case, this is outside the space. That's why when it fused here, it was secreted to the outside. Now this one is not fully secreted here, it's bound here. This part is exposed to this space. This is this space. Which space is inside? This is inside, right? This is outside space. If this whole membrane moves to the plasma membrane and fuse here, then this side should be exposed to the outside. C terminal should reside inside. If you are not clear, then you can make this into a small ball, and then that ball is moving to the surface, and then try to fuse it. Okay. Anyway, outside connected to outside, inside connected to inside. Then this is residing inside. C terminal is inside here, and terminal is exposed to the outside. Okay, if it is not super clear, then you practice with this. Then definitely you will see this problem again very soon, maybe in midterm exam or final exam. This is quite important the fact. If this is not clear, then maybe you may handle this later again, but you can figure it out, I think. Now, coming back to the mitochondria. If this protein is just delivered to the surface of mitochondria, this is outer membrane of mitochondria, inner membrane of mitochondria, okay? And this is inside, this is inside, this is outside the space. One possibility is just this protein is bound to this surface. Then this protein is exposed to the facing cytosol. That's one way. There are many different kinds of mitochondrial protein. They may bound to the outside surface and then just facing to the cytosol, one way. There may be another carrier which can move this through this channel, move this protein through and entering into matrix area. That's also possible. Then this whole thing goes in, and then they are inside the mitochondria matrix protein, they may be. If the channel is here, not connecting these two membranes together, maybe channel is here, okay? Then this protein can go through and then just secreted into this outside the space. This is intermembrane space. That would be also possible because all the side here, 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 they did the proteins, different kind of proteins. Okay. Now the role of a Hisha protein is rather, the two passes through this small channel. Structured three-dimensional structure may not be easy to go through. Outside here, this Hisha protein just make this reopen and then let them go in. After they pass through this small channel, they need to reform the three-dimensional structure. So then you can assume that this Hisha protein has a reversible reaction. Sometimes it's opening, or sometimes it's folding. Okay, so three-dimensional structure cannot go through this. That's why I make it linear, open, and then go through. And then inside here, restructure those proteins. That's what is happening here. Okay, so our time is up. So let's finish today's lecture here. Let me go back to my slide. Okay, next week class is the DNA to RNA, RNA to protein, and also important the technique of DNA sequencing. Okay, so now we learned some the macro view of those molecules, how they are synthesized and how they are localized into which part of the cell. Now from now on, we are handling a little more molecular level, and how the coronavirus is diagnosed by this technique, kind of DNA sequencing, and also those translation, transcription will be covered next week. So same way, next week, Tuesday, you will answer those quiz problems. And then assignment 6 will be same way, the Wednesday, 6 a.m. And if you are late or any other reason, you need to modify those structure, then you may send me by email. Okay, any urgent question for today's class? Okay, if not, so let's finish here. If you have any difficulties in the middle, please communicate with me as soon as possible. Okay, if you give me your the notice at the last minute, then I have to hurry up to rechange those plans. Sometimes it's quite difficult for me. Okay. Okay, guys. Thank you, and see you next week. Bye bye.
Thank you, Professor.
Thank you.
Thank you,
Thank
Professor.
you.